I'll just record it. So I'm recording a tutorial on DNA. We've covered the inheritance of 23 chromosomes from each parent. Chromosomes are condensed DNA, showing the chromosomes here. Humans have 23 pairs, 22 of those are autosomal, and the 23rd pair are the sex chromosomes that determine the sex of the individual. So alternative forms of the gene are called alleles. So this is one allele here. Let's call that the brown allele for eye color. And this is another allele, the recessive allele. And they're at a very specific location. At a specific location called a locus. Whoops. If there are several, the plural form is loci, different locations. Are you with me so far? So, good, okay, awesome. All right, those are alleles, very specific locations. So what happens in meiosis, and I'm not going to go over meiosis in detail, but it's like mitosis, which you do need to know mitosis, but it's like mitosis, but it's like the cells split twice. So in meiosis 1, the first split, what happens is all of your DNA before this split have been replicated. So you've got 2 copies of everything. Oh, one thing I wanted to mention also is that these 2 chromosomes, before I go on, are homologous. You have 23 pairs of homologous chromosomes. And they're called that because they are similar, but not exactly the same. It's like your shoes. Okay, hang on. Let me find my shoes. So when I'm in the class, I make students do this. It's like, lift up your shoes. So your shoes are similar, but they're not exactly the same. One is left, one is right. One is like much dirtier on the bottom than the other one, because I've been walking through mud for the last few months. So they're homologous shoes, okay? They're similar, but not exactly the same, just like homologous chromosomes. When you replicate a chromosome, in this case, I've replicated the chromosomes here. Those are called sister chromatids. And I'm just telling you that. You'll need to know that when we get to development. At the moment, you don't need to know that particular fact. But what I wanted to show you was in meiosis, the first division is the division of homologous chromosomes. So in the first division, you'll get 2 cells like that. And in the second division, the sister chromatids divide. Like so. And then when it comes time for For fertilization, those chromosomes will come together again. So what these are here are gametes. They are either an egg or a sperm, depending on whether or not, of course, the person, male or female. And they'll have, in the end, of course, as a human, since we have 23 pairs of chromosomes, each gamete will have 23 chromosomes. And that's really important because if the eggs and sperm had all 46 chromosomes, well, every time they came together in fertilization, you'd have double the number of chromosomes. So you have to have the number of chromosomes during meiosis. And that way, when fertilization happens, you'll have the same number, 46. And it will be stable or maintained throughout generations. So when you're looking at fertilization, you're doing what's known as a cross. A cross. So in this case, our adult has the genotype BB. That's the genotype. Big B, little b. I'm going to say big B, little b. So we'll just look at one chromosome at the moment. Once you start looking at 2 and 3 and 4, oh my gosh, we could look at 23 chromosomes inheritance. That would be crazy. And the reason I say that is that it's a lot of chromosomes. We're only actually going to look at one gene on one chromosome. Yeah, if you were a geneticist, you'd be looking at, well, 23,000 genes on 23 chromosomes. Holy Toledo, that's a lot. But the thing is, there is a probability of getting a particular allele. And the probability depends on what your parents have. So if this parent has a large B there and a little b there, and makes gametes, I'm going to skip the stages in between, but the gametes, we've only got two possibilities, right? You can have this chromosome or this chromosome. You can get B or little b. What is the probability of getting big B? What is that probability? What are the chances? Just like flipping a coin. 50%. Right, so you have 0.5, that's 50% in ratio, proportion, and 50%. Yeah, so it's good to work out the probability of the gametes. Now, if we were to work this out, for chromosomes that you have inherited, I don't know if I told you this before, I probably did, but since there are 23 chromosomes and, one egg might get this one, the other egg might get this one. But if you have, we're looking at two different chromosomes, for example, like that, with their homologous partner. These will split into different gametes. Well, you could get 4 possibilities here. You could get this one, this one, this one. Yeah, this one and this one, or this one and that one. Get this one and that one, or this one and that one. So there you've got 4 different possibilities of gametes, 4 different combinations of those 2 chromosomes. And it turns out that for you to get the exact combination of chromosomes that you got from your mom, that's 1 in 8 trillion. To get the same combination of chromosomes from your dad is also 1 in 8 trillion. We have to multiply those together because the probabilities of getting one thing and another thing is always a multiplication. That's 1 in 64 trillion. So the probability of you having those chromosomes is 1 in 64 trillion, and that's how unique you are. And that doesn't even take into account the probabilities of your parents even meeting or having sex on that particular night. Sorry, but your parents did have sex on one night in order to have you. Anyway, that's kind of an aside. But we're only going to be looking at one chromosome with one allele. So we said that this is the genotype of the individual. Sorry, I'll go back to black here. Now, I always like to write down what the alleles code for. So we know that big B, in the case of eye color, is brown eyes, and little b is blue eyes. A dominant trait will always be expressed if it's present, and that's the dominant trait there. Dominant, recessive. You must have 2 recessive alleles to express that trait. So if you have the alleles, that one or that one, these are the genotypes. You'll have brown eyes. That is the phenotype. Phenotype is really just another word for the physical expression of the gene or allele. So those are genotypes and phenotypes. Wow, everybody's still here. Almost. That's amazing. I was expecting like 1 or 2 people. Okay, but I'm so glad that you stayed. That's lovely. It makes me feel very valued. Okay, so are you with me so far? Genotype, okay. Phenotype, okay. Good. Okay, lots of thumbs up. That's awesome. Okay. Good, good. Now we get to the question of, well, what are my offspring going to have? What will they inherit? What, in particular, as a genetic counselor, people are going to want to know, what is the probability of my offspring inheriting this particular trait? Well, let's start really simple. Let's start with the monozygotic cross. I'm going to stick with eye color, even though eye color is actually more complicated than this, but it's kind of good to go with it anyway. There happens to be more than one allele for eye color. There's a green as well, but let's just go with this for now as an example. It's a good example to use. So now we're going to do a cross, a monozygotic cross. To see what we'll get in our offspring. one gene, one trait. So we always have the parent population, the parents themselves. Big P, that's what we call our parents. And we're going to look at their genotypes. We're crossing them by genotype. So the genotype of one parent in this case is homozygous dominant for brown eyes, and we're going to cross with a homozygous recessive individual of blue eyes. So the first thing we want to ask, what are the possible gametes these individuals can make? Well, this individual only has one type of allele, so the gametes will only have that one. This individual also just has one. So these are the gametes, and I always like to draw it out because it makes it make more sense to me. So well, now, of course, these individuals will go through fertilization. And when they're fertilized, what happens is they fuse. So fertilization equals the fusion of the nuclei and the chromosomes in the nuclei come together. So what do we have in our offspring that have inherited these traits? That's the F1 population. The big F stands for familial. So that's our first generation after our first cross of the parents. And we know that they're all going to be heterozygous because that's the only possibility. They've got a big B from one parent, a little b from the other parent. That's really all the possibilities that they had. So that's the F1 population. But now we want to know, well, what will happen in an F2 population with crossing one individual that's heterozygous and another individual that's heterozygous? What probability is there of alleles in different gametes? Well, for this individual, one gamete could have a big B, the other one a little b. For this individual, one B and one little b, because these alleles are in chromosomes, and those chromosomes have been pulled apart into different cells that are eggs or sperm. So that's a little bit more difficult to assess. That's why you use a Punnett square. I'm going to go to the next page. This is bigger. F2. It's easier to figure out what the probabilities are if you do a Punnett square. It's easier to understand them. So my question to you is, what is the probability of this gamete getting the big B and this gamete getting the little b? What are those probabilities? It's like flipping a coin. Same as before, 50% exactly. 50%. Same with the other parent. Remember, these are the parents. This is mom. This is dad. So the reason we do the Punnett square is we say, okay, well, this individual will be the gametes from the dad. One of them could be a big B, the other one could be a little b. We don't know. We don't know. It's random. Whichever sperm gets together with the egg is random. So we want to know the probability. Yeah. So from the from the mom over here, from the egg, well, we've only got these two probabilities, right? And so the Punnett square is there so that you can combine them easily. So in this square, I'll put the big B's together, big B, little b. You're just combining them into the appropriate boxes here. And this one, we'll put the little b and little b together here. Yeah. So it's nice to look at a square because you know that it's even. So we've got 4 boxes. So what's the probability of getting any one of those? So there's 4 squares. What's the probability of 1 square? A quarter. Yeah, so 0.25, 0.25, 0.25, 0.25. Another way of looking at that is that the probability of getting this B was 0.5. The probability of getting this B was 0.5. This is during meiosis. Probability of that one was also 0.5 on the mom side. The probability of that one was 0.5. So how do you get this probability any other way? You just multiply these together. 0.5 times 0.5 is 0.25. So the probability of getting an offspring with the big B is 0.25. The little b is 0.25. The big B, little b is a bit different because you can get it 2 ways. You can get the little b from, mom or dad. So if the probability is one or the other, you add them together. And that makes sense. If you have a coin and you flip it, ding, you go, is it going to be heads or tails? Well, there's a 100% chance of it being heads or tails, right? So you add those together. 0.25 plus 0.25 is 50%. So then in the end, you have BB 0.25, big B, little b 0.50, and little b 0.25. And so the ratio is 25, 50, 25. You can write it like that. You could write it, I wish I'd done them side by side. You could go 0.25 to 0.50 to 0.25. You could go 25 to 50 to 25, but you could also go 1 to 2 to 1 as it's a ratio. Good, yeah, 1 to 2 to 1, exactly. So that's the ratio. So let's go back to similar questions that were in your handout. What is the genotype of the F1 population? What is the genotype of the F1 population? I'm looking at the chat. Yeah, the genotype is big B, little b. All of them were that. What is the phenotype of the F1 population here? Brown eye color, good, yeah. Excellent. And of course, these are all genotypes. Genotypes are just the combination of alleles. But the phenotypes are brown, whoops, brown, and blue. So the phenotypic ratio is different. The phenotypic ratio is 2 to 1, 3 to 1. Sorry, because you've got this 50% here. Yeah. So good. I think we've answered all the questions in that handout. Are there any questions from the audience? That's you. Not so far? Okay, good. Excelente. So do you have questions about, I have a question here. Are the questions on the exam going to be like the ones on the worksheet today? You won't have to work them out. No, but you'll need to know what all the terms mean. I don't know if there will be a short answer question. I have not decided yet. But if there is a short answer question, then it would likely be like this question, yes. So do some practicing, yeah. But I won't I don't think I'm going to do a dihybrid. We're not doing dihybrid crosses. Yeah, just monohybrid crosses, like this one that we just did. I hope that cleared things up for you. So far, so good. Trying to think of what else to lecture on. Hang on. Let me